So to the main business of the evening. As you all know, our health has very much dominated the news over the last 18 months, and we haven't even been able to go out and get a pint of milk without having to consider and mitigate for the risks to ourselves and to those around us. But one of the positives that has come out of the pandemic has been it's generated a lot of interest in science, and in particular in the important contribution that our genes play to our health. And this is something that our scientists at the Epilepsy Society have been studying for many, many years, obviously in relation to epilepsy. And as non-scientists at the Epilepsy Society, we're often privileged to hear Sanjay explaining about genetics and the fundamental mechanisms that contribute to our health. And it's always very exciting and very inspiring. And so we thought, well, Chalfont St Peter is pretty unique in that it has a world-leading research centre right in the heart of its village. And so we wanted to share with you that the privilege of the expertise that we have on your doorstep. So with no further ado, it is my great pleasure to hand you over to Professor Sisodiya.
Thank you very much, Nicola, for that very kind introduction. So I'm just going to share my screen. Okay. Can you see that? Nathan, thumbs up? Yes, no?
Yes.
Yeah, okay, great. All right. Well, thank you very much, Nicola, for the very kind invitation. And thank you to all of you for giving up your Wednesday evening to listen to a talk about science. So I'm going to be talking really about how science is making an impact for people with epilepsy. And as Nicola says, I think science has really shown us the way forward. This year, we all know about this. It's affected every one of us in some way or other. And I think what's really striking, apart from the huge impact it's had on everybody's lives, is how quickly we went from knowing that there were a few people who had an unusual illness to then seeing this spread across the world, and then very rapidly identifying the problem, identifying the cause of this illness, which is this virus. This is seen at huge magnification. And very quickly after this was identified, its genes had been studied completely. So we know its genetic code completely. And that's what has allowed us to make these other steps where we can track and trace the spread of the virus, and of course, develop vaccines against the virus. And this has really shown the power of science, and I think it's brought it home to all of us, what exactly it can achieve. Now, I'm not going to be talking about science everywhere. I don't know about science everywhere, and I don't know much about viruses either. I know a little bit about epilepsy, and so that's what I'm going to focus on. And as Nicola says, this is not only science made simple, but also science round the corner. This is in your backyard. So this is a map of our area. Here's the 413. There's the Epilepsy Society here, the Chalfont Centre, and this is an aerial view of our centre. And so this is where the science takes place, and my office is somewhere here. And this is where we see people with epilepsy, especially people whose epilepsy is difficult to treat. And we try and use the science that we have at our fingertips to help with the problems that they have. So the Epilepsy Society has been around in Chalfont for a long time. It's founded in 1892. Here's a very early picture of the society. It was founded by a group of neurologists, doctors interested in the brain, and it's still going strong. Here's one of our latest editions. This is our research centre. And this is where a lot of the science that I'm going to talk about takes place. So I'm sorry if you've just had your dinner and I'm going to show you this picture, but it's all about the brain. So what is epilepsy? Well, it was around about the time that the society was founded that a group of neurologists in London, led by this gentleman, Sir William Gowers, worked out that epilepsy was a disease of the brain. And here is a picture of the brain This is someone having surgery for their epilepsy. Their epilepsy has proved very difficult to treat. And so we're looking here to see whether we might be able to remove the part of the brain that's causing the seizures. So epilepsy is a disease of the brain, and it's characterized by the occurrence of seizures. Now, this might be familiar to many of you, but perhaps not so familiar to others. So I'll just say a little bit about this. So in epilepsy, what happens is that people continue to have seizures. The seizures are out of their control, and the seizures can prove dangerous and require treatment. And the seizures come from the brain. So here's a schematic picture of the brain. You can see the wiggly surface, which you see here in real life with the arteries and the veins on its surface. And the brain is made up of lots of different cells, and the cells are made up of smaller and smaller components. And chief amongst these are the proteins. These are the actors in the cells that actually do things. And these proteins are generated in turn by our genetic material, our DNA. So epilepsy is characterized by seizures. Now, probably many of you will be familiar with the most serious sort of seizure, when people will fall over, they won't be responding, and they might have stiffness and jerking of the limbs. And this is something that we've all seen that used to be called a grand mal seizure. We now call them bilateral tonic-clonic seizures, which describes what happens with the limbs becoming stiff and jerking. And these are very unpleasant seizures to experience, and they're dangerous. But actually, seizures can take lots of different forms. That's the most dramatic form, the form that I think we could probably all recognize. But sometimes a seizure is just a period of absence. Someone stops what they're doing, may look blank, won't be responsive, and then can come back and join the conversation as if nothing had happened. And you get lots of other sorts of seizures which vary in severity between those two. So epilepsy is characterized by these recurrent seizures that are out of the person's control. So another way of looking at this is that it's an imbalance in the function of the brain. Now, in the brain, we have two main processes that go on. One is called excitation, making things happen, and one is called inhibition, stopping things from happening. And in the brain, there's a fine balance between excitation, making things happen, and inhibition, stopping things from happening. And a lot of science has gone into an understanding of what happens in seizures, of how this balance between excitation and inhibition, between excitation, if you like, the accelerator, and inhibition, if you like, the brakes, how those two work together. So imagine driving a car, you need an accelerator and you need brakes. You need both to be able to drive the car safely. And in the same way, the brain needs to be able to balance these two things from happening. So let me just try and illustrate that. Okay, so this, if you like, is a balanced function. This is something that's happened in a controlled way. Okay, so this is me drinking a glass of water. Okay, it's all very balanced. It's under my control. Excitation, inhibition, both working together so I can get the drink of water. Okay, now imagine the same thing, but here you see someone's hand. Okay, and this is someone else who's controlling my hand. So I don't know what they're doing. I don't know if they're going to pull it, stop it, let it go. I don't know. Okay, so this is no longer balanced, and you can see obviously what happens. Okay, I'm trying to get this drink. It's my son being very difficult. He's letting me do it? No, he's not letting me do it. And of course, I make a mess. So I've made slightly light of this, but this is basically what's happening in the brain. There is excitation and there is inhibition, and the two need to be balanced. And if they're not balanced, then people have seizures, and when they have seizures, then we say they have epilepsy. So it's a disease of the brain, and there's a lot of science that's gone into working that out. There's also a lot of science gone into thinking about what causes epilepsy. So it's a disease of the brain. You can imagine anything that affects the working of the brain could cause epilepsy. There are lots of different causes. Now I'm going to show you some causes where you can see in the brain directly what's going wrong. And to do this, I just need to show you this picture first. Okay, so this is just to illustrate or explain the next slide. So here obviously is a loaf of bread. Imagine this is the brain. It's the brain that is the seat of the epilepsy. It would be brilliant to be able to look inside the brain. Okay, and one way you could look inside that, obviously, is to cut it open and have a look. And so you can see both cut faces of this loaf of bread, and you can see actually there are some olives in this. It's olive bread, okay? Of course, you can't do that to people. You can't go around slicing the brain open to see what's in there, but you can do the next best thing, which is to virtually slice it open. We're very used now to virtual things, virtual meetings. This is a virtual meeting. So you can virtually slice the brain open, and you can do that using a scanner. And some really, really clever science went into working out how to make a scanner and how to make it look into the brain. And actually at the Epilepsy Society, we were the first place actually in Europe to get a scanner just for epilepsy to look into the brain. So here are some, if you like, cut faces of the brain. So these are images of the brain from different people, 8 different people who have or had epilepsy. Okay, obviously we can't look into their brains directly, but we can look into the brain using a scanner. And so you get these pictures of the brain. Okay, so here you've got a picture of the brain, and you can imagine it's basically like cutting into this loaf of bread. This is really clever science. Okay, so some really clever scientists made these machines, and we use them now at Chalfont. And so you can see that for some people with epilepsy, there are changes in the brain. So here you can see something very obviously going on in this part of the brain. This, unfortunately, is a brain tumor, and we know that brain tumors can cause epilepsy. It doesn't mean everyone who's got epilepsy has got a brain tumor, but some people who have epilepsy and suffer seizures have a brain tumor. Here's another different sort of brain tumor, a more benign sort, but that can still cause seizures. Sometimes you can see an area of scarring, perhaps as a result of a head injury long ago. Sometimes you can see some unusual things. This is, if you like, a birth defect in the brain. This is a bit of brain tissue, the grey matter of the brain, that should really be on the surface of the brain here, but never quite got there. And this can cause epilepsy. And of course, people can have strokes. This is what a stroke can look like on a scan of the brain. And this stroke would have affected this person's ability to use their body and might also cause seizures. So there are some obvious causes that you can see on a scan of the brain. And the scientists at the Epilepsy Society who work on the scanner are always tweaking it to try and get better and better pictures and find more and more causes where you can actually see something going on in the brain. But sometimes you can do a really, really good scan of the brain and still not see anything. It looks completely normal. That could be because the scan's still not good enough to show what the cause is, or it could be because the cause just won't be seen on a scan. It's something else. It's not something you can see on a scan. And how do we treat the seizures? How do we treat the seizures that happen in epilepsy? It's important usually to treat them because the seizures are unpleasant to have. They stop people doing things, for example, like driving. People might fall over, hurt themselves, break bones. And even very rarely, seizures can even threaten life. So it's important that we treat them. And we've been doing this for a long time now at Chalfont. So here is a record of seizures from 1926 from somebody who lived at the Society. And what they did was to record each seizure every day. So for example, here's April the 1st. This unfilled triangle is the daytime, the filled triangle is the nighttime. Every time this person had a seizure, either in the daytime or the nighttime, it was recorded. And this person got various treatments for their epilepsy. In fact, this person got something called potassium bromide and got a few grains of this. This is how it was measured then. And this is a drug we still use. We've got a lot more drugs now, and this is again science. Science has brought these new drugs and made them available to us. And so we have something like 25 different drugs now that we can try to bring the seizures under control. And the science has been good here in bringing these drugs to us, but I have to say that for most of these drugs, we don't really know exactly how they work. We have a rough idea for most of them, but we don't know exactly what they're doing in the brain. And what we also see is that for about 1 in 3 people who have epilepsy, all the different drugs we've got don't work. They don't stop their seizures. So people continue to have seizures and have all the risks associated with them. And this is where we need more science to try and work out what's going on for those people and see how we can bring their seizures under control. And that's what I'm mainly going to be talking about in this talk. So I'm going to be talking about somebody called Charlie. It's not his real name, but it is a real story. And here we go. So Charlie is 19. He's had epilepsy for his whole life. He has lots of seizures and different types of seizures. So not just the convulsive seizures, but also other different types of seizures. As well as this, as we often see for people whose seizures are frequent and difficult to treat, he has other difficulties. So he has problems with learning. He's never been to a school, a normal school. He has problems walking. He has problems speaking. And the problem is, oh, sorry, I beg your pardon. You're wondering why this woodpecker is here. And that's really important, actually. I'm sorry I missed that. That's because, of course, that epilepsy doesn't define Charlie's life. Charlie has lots of interests in his life. And one of those interests is actually the birds that come to his bird feeder and sit at his bird table. And he'd really like to be able to get out and see more birds and wildlife, but his seizures often stop him. The problem is that he's one of the 3 people for whom the medications we have aren't working. This means that the seizures continue to happen and get in the way of his interests. It also means, unfortunately, that his walking and his talking, which are already impaired, are deteriorating. So what's going on? What's happening in Charlie's brain? Can science help us understand this and maybe help us treat Charlie's seizures and his other problems? So of course, when the drugs aren't working, it's really important to try to see why somebody's got epilepsy in case there's another way to treat it. So Charlie's had lots of tests for his epilepsy, try and identify the cause. This is a scan of Charlie's brain. And you can just about see here, for example, the ears, okay? So this is like that loaf of bread I showed you earlier on. I can tell you this scan is normal. So we can't see an obvious cause in his brain, but there must be something because Charlie has really difficult to treat seizures. So this is when we thought, well, maybe Charlie's seizures are caused by a problem in his genes. Okay, so you all know about genes. Genes are largely what make us what we are, not completely. We're not entirely determined by our genes, but they largely make us what we are. We get our genes from our mother and father, of course, half from our mother, roughly, and half from our father. Okay, so Charlie's got genes, and his genes have come from his mum and his dad. The brain is a really complicated structure in the body. It's a really complicated organ. It's made of lots of different cells, okay, like a big Lego structure made of Lego bricks. And these cells, these brain cells, are themselves made up of other things. Eventually, they're made up mainly of proteins. And all the things that the cells that make up the brain are themselves made up of are all determined by something called genes, the genes that I've been talking about here that we get from our mother and father. And the genes themselves are made up of a chemical called DNA. I'm sure you've all heard of this, and there's been a lot about this in the news recently, especially related to the pandemic. So this is how the genes are made up, just a quick summary. So the DNA is the genetic material. It's made of these things called base pairs. We represent these as letters, G, T, C, and A. So it's just 4 letters, but these letters can be arranged in lots and lots of different ways. So even if you've just got 4 letters, you can arrange them in many, many different ways. You can imagine you could have G, T, C, T, 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 C, T, A. That's one way of arranging them. But you could also make that G, C, C, T, T, G, C, A. And you can arrange it in many, many different ways because there are lots of them. So these base pairs that make up our genetic code are the DNA, and the DNA is organized into bigger units called genes. And these genes are packaged into chromosomes, and the chromosomes are, if you like, the headquarters, if you like, of the cells that make up every bit of our body. Okay, so this is the boss part of the cell. This is where all the instructions come from. And the key unit in our genetic material in all of this is the gene. Okay, so here's another way of thinking about this. You can think about our genetic material, and if you think about all the genes together, we call that our genome. Okay, so you can think of our genetic material, if you like, like the script of a play, okay? And as you know, the script defines the characters, tells us what they're going to do and what they're going to say, okay? So it's the instruction manual, if you like. And then the proteins in our body are like the actors. They're the things that actually do things. They're the things that, for example, are working as I'm talking to you. They're active in your eyes as you're watching this. They're active in your ears as you're listening to this, okay? They're called the proteins. They're the actors. They're the things that do things. And they're made up as a result of the activity of our genetics, of our genes. So coming back to think about Charlie, okay? So Charlie's brain is obviously not working as it should because there are lots of seizures happening and there are other problems happening, all related to the brain. Charlie gets his genetic material, obviously, from his mother and his father, half from his mum, half from his dad, okay? So the question we had in mind was, might there be something not quite right with Charlie's genetic material that's causing his epilepsy, okay? So now we're beginning to apply our scientific knowledge of genetics to trying to understand what's wrong for an individual person, okay? So all our genetic material, which is all this DNA, is made up of 3 billion of those letters, G, C, T, and A, all organized, all very highly organized, and all in a particular order. And that's our DNA, 3 billion letters. And science now has enabled us to read all of those 3 billion letters. That's a really amazing thing. When we first did this, and I don't mean me or at Chalfont, but when scientists around the world first decoded the human genome, it took them more than 10 years and it cost a huge, huge amount of money. And science has now advanced to the point where we can read all 3 billion letters in less than a week. And it doesn't cost $10 billion, it costs more like £1,000. It's still a lot of money, but it's much less than it was. And this is all as a result of the advance of science and technology. So this means it's really now realistic to read one person's entire genetic code, all 3 billion letters. And we do this regularly at Chalfont when we're trying to work out what the cause of somebody's epilepsy might be. So one way to do this is that we read all of Charlie's 3 billion letters, and we read all of his mum's 3 billion letters and his dad's 3 billion letters. And if you remember, Charlie's 3 billion letters in his genome have come half from his mum and half from his dad, okay? So that everything Charlie has should be seen either in his mum or his dad. And what happens, of course, when Charlie is formed is that the DNA in his mum is copied, half, into Charlie, and DNA from his dad, half of that is copied into Charlie. So there's a process of copying that goes on, okay? Now imagine you had to copy 3 billion letters, or 1,500,000,000 from each parent. That's a lot of copying. And it wouldn't be surprising if you were copying that and you made the occasional mistake. In fact, you probably make a mistake in 100 letters, let alone 1,500,000,000. So mistakes happen. And these mistakes that happen, we call mutations, or probably a better word for them is variants. We all have these because this process of copying, although it's fantastic, still gets it wrong every now and then. Okay, so I've got a few variants, you've all got a few variants, we've all got them. That's part of human life. And of course, sometimes those variants help us, but sometimes they don't. So if we come back to this analogy of the script being our genome and the actors being the proteins that actually do things, we've got about 20,000, let's say 20,000 paragraphs, I don't know, let's say in Shakespeare's, in one of Shakespeare's plays. That means there are 20,000 actors. That's a huge number of actors, all working closely together to allow us to do all the things that we do, like generating vaccines against COVID. So we can do fantastic things as a result of this complexity. But you can imagine if something goes wrong, that could be a problem. Imagine in this script, there's one wrong letter, okay? So here is an example of this. You all know this, of course, alas, poor Yorick, I knew him. Okay, imagine if there was a mistake and that K was missing. Alas, poor Yorick, I knew him. So for the person who's reading this, doesn't make sense really, but of course, if you're listening to it, it doesn't make any difference. It sounds exactly the same. So this is a mistake, okay? You can imagine this could be caused by a single wrong letter in the script, but this mistake doesn't really matter. No real consequence. On the other hand, imagine a mistake like this, a stage instruction, exit stage left. Okay, we're all familiar with that sort of phraseology. But imagine if this I was miscopied as an A and it said exact stage left. That doesn't make any sense. That could cause a problem. If you were the actor reading that, you think, what am I supposed to do? So obviously we'd probably kind of make it up, but you can imagine how this could cause a problem. So this is what happened for Charlie. We read all of Charlie's genetic code, his entire genome, and that of his mum and dad, and we looked for the differences. And we found Charlie did have a few mistakes, a few copying errors in his genetic code. Most of those didn't matter, but one of them did. Just one caused a problem. And what that mistake did was that it told the protein, the actor that it produced, to be too busy. Okay, so here's a little graph. It just shows you this is how busy that protein should be with the correct letter. This is normal, okay? With a mistaken letter, that protein, that actor in the brain was too active. It was doing too much. And you've seen how in the brain you need this balance. Everything has to be in balance. You have to have excitation and inhibition, the accelerator and the brakes in balance. And here you've thrown the system off balance. And this single letter that was miscopied is the cause of Charlie's problems. So let me show you that in a more scientific way, okay? So this is a cartoon of that protein or the actor that's gone wrong, okay, in Charlie's case. These letters and numbers represent different bits of this protein, and this protein sits in every cell in Charlie's brain, okay? And it makes a lot of those cells behave incorrectly, and that's what leads to Charlie's epilepsy. Okay, so this is another way of looking at that. What you can do is you can actually make this protein in a dish, and you can make the normal protein here and here, and you can make the abnormal protein that's got that single wrong letter. And you can see this is how busy this protein is, okay? These lines just represent how busy it is. It should be about this high, its busyness. But in the protein that's not right because of the wrong letter, you can see it's much, much more active. It's doing too much. It should be doing this. It's doing this. Now, it just so happens that as a result of other science, we happen to know that there's a drug that we never use for epilepsy. There's this drug which happens to fit in this protein and which affects the way it works. So here's the normal protein, okay, studied in a dish, working away normally. If you add this drug, you suppress the activity of that protein, you damp it down, okay? So you don't want to do this for healthy people because you don't want this to happen, you want it to be working normally. But if you've got a protein that's too busy, you see, you reduce the activity of that protein back down to normal. This here looks like it should look like, not what it would look like otherwise. So here we've got a drug which exactly treats the problem in Charlie's brain. Exactly. Okay, we call this precision medicine. This is not using a blockbuster drug that works for everybody in a way that we don't really understand, but most of the time is very effective. Those drugs aren't working for Charlie because they're not targeting the problem. Here, we've got a specific drug that works exactly for the problem that Charlie's got. It reverses this overactivity and makes it normal. So we would hope that this drug would work for Charlie. Okay. Now, Charlie's condition, which we now know exactly, we don't have to call it bad epilepsy with learning problems and walking problems and talking problems. We can give it an exact name. And the exact name tells us what the problem is. It's actually called KCNA2 gain of function, developmental and epileptic encephalopathy. It's a real mouthful, but it tells you exactly what's going on. And what he's got is a really, really rare condition. I only know of about 20 people across the entire world who've got this condition. There are probably more, and we don't know about them yet because not everywhere in the world has this science. They can't all access this science. But in places where they can, and people have studied it, we know there are about 20 people with this condition across the world. And so other doctors like me and scientists around the world have all got together to try and understand this better, to use the science to try and help. And that's led to the discovery that that drug I told you about can reverse the effect of that overactive protein. And so there are some children around the world and other adults around the world who've already been found to have this problem. We've only just found it for Charlie, but others have been known to have this condition for a longer period of time, and they've tried this drug. And for most people who've tried it, it's stopped their seizures, it's improved their walking and improved their talking, where no other drug has worked. And so the science has taken us from not understanding why these people had epilepsy to a really detailed understanding of what's going on in their brain, all through science, and the discovery of a drug that could work for them, and for some children has worked. And this is really important because although this condition is really, really rare, it just so happens that I also see another person with this condition. He's much older, and he never had the chance really to benefit from all of this science, because when he was younger, this science didn't exist. It's only much more recently that we found that he has exactly the same thing as Charlie. And unfortunately, my older patient with this condition still has very bad seizures and no longer walks and no longer talks. And we've missed the boat there. And that's really sad. There's nothing we could have done about it because the science didn't exist then. But what it shows us is we really need to work out what's going on in each person with epilepsy, because there might be a way that we can really stop what's going on, stop their seizures. And so what we want to do for Charlie is to use this drug. So we're just about to start this drug for Charlie. And I really hope that we can stop that progression where his seizures get worse and his walking deteriorates and his talking deteriorates and he ends up in a wheelchair and not able to do anything for himself. We really hope that by using science, we can find the right drug and therefore help him. Now, Charlie's condition is very rare, but Charlie's story is not. There are lots and lots of people who've got bad epilepsy, and in many cases, that bad epilepsy is because of a genetic problem. And some of those people might have a genetic problem for which there is a specific treatment available. And so people come from all over the country. These purple dots represent where in the country people come from to come to us at Chalfont. Okay, so Chalfont, as you know, is around about here. And you can see people come from all over the country to our centre. And we try to use the best of science, including scanning the brain, including studying people's genes, to see if we can find out why people have got epilepsy and how best we can treat them, how we can stop the seizures, and how we can stop the other problems that sometimes happen for people with epilepsy. So although Charlie's condition is really, really rare, there are many, many people who've got epilepsy like Charlie's, which is serious and difficult to treat. And so we're trying to use these methods and these techniques in this science to help many more people with epilepsy. So I'll stop there. I don't know how I'm doing for time, but I'll stop there, I think, and happy to take questions.
Lovely, thank you, Sanjay. That was really illuminating. I'm just wondering, is anybody brave enough to ask any questions? Please do feel free. They can be as basic as you wish, or as clever as you wish. Liz.
I was just wondering, for Charlie, you've actually found, this possible drug that he can take. You also mentioned the other gentleman who was much older. Is it not possible to give him the opportunity to have this drug as well?
So it's a very good question. And of course, what I've tried to give you today, I suppose, are the broad ideas and the broad concepts of the sorts of things we're doing. But you can imagine that there are lots of nuances and complexities, which I haven't really had time to go into this evening. Sure. So in fact, the person I see who's much older than Charlie, who's got the same condition, has tried this drug. We did try it for him. It was really difficult to get. It was really, because that was a little while ago. And unfortunately, it didn't help him. He's the only person in the world I know for whom this drug did not help. And I think it's because he's the oldest person in the world I know who's got this condition. And I think we've missed the boat.
Fine. Thank you very much. Thank you.
Thank you, Liz. We've also got a question in the chat from Exima Aziz, who asks, what is the process when patients are admitted into the centre?
So the way this works is that so the person is being referred by their GP or by their local neurologist. We get a letter of referral, and then we see them in our pre-admission clinic, just to find out about their problems and to make sure that we can cater for their needs, and they get to look around our centre and look around the admission unit. And then we arrange the admission. They come in and we address, if we can, the problems that they might be having with their seizures or memory or other problems. Or we try and work out if they have got epilepsy or not, and what other problems they might have. That evaluation can be as short as 2 or 3 days sometimes, and sometimes it can be a longer admission where we're also changing the drugs during that admission. So it varies very much from person to person.
Thank you. We have another question from Yasmin Golding. Yasmin, would you like to ask the question yourself?
Yes, I'm here with my mum today, and she'd like to know if Charlie has to take any other AEDs, or if that medication is precise enough to treat it alone.
So it's a really good question. And of course, what we'd like to do, if we can, is to control Charlie's problems with the specific treatment, and then try and get rid of the other drugs if we can. And that's what we will try to do. But of course, we don't know if that'll work or not. And we have to try that step by step. So we'll bring in the new drug first. And if that suits Charlie and it works for him, then our plan will be to slowly wean away the other drugs one by one. That will be the plan. But things don't always go to plan, of course, as you know.
I do, thanks.
I think somebody was, yes, Peter Powell. Would you like to unmute yourself, Peter?
Yes, thank you. That's most interesting. I just wondered if you anticipate that there might be an opportunity to intervene at the gene level if you detect this condition obviously very early.
So people are working on that. It's a very good question. And in some cases, it might well be possible. In other cases, it might not be possible. It depends actually on the particular gene and how it's leading to the seizures. So it's not possible to say, I think, with a broad brush that yes, gene therapy would work for everybody. But for some people, for some types of epilepsy, it might well work. And there are some trials starting up for gene therapy for particular types of rare genetic epilepsy.
Thank you.
What we don't know if it would correct everything, because you only find all this once the problem has started.
Sure. Thank you very much.
Is that Santha would like to ask a question?
I have a question, Professor. This treatment, what you have mentioned, is it effective for all this type of gene mutation type of epilepsy, or any other type of encephalitis, like syndromes, like Rett syndromes, like Rasmussen encephalitis, or something like that, or particularly for this gene mutated epilepsy? How?
It's only for this epilepsy, not for any other epilepsy. It's very specific, and it's what we call a precision treatment. So it's the right drug for people with this problem. It would be completely the wrong drug for anybody else.
All right, thank you.
Okay.
And then we have Walter. Would you like to ask a question?
just a question. So is there a case for everyone who is newly diagnosed with epilepsy to be genetically profiled?
Well, again, a very good question.
Whenever diagnosed, whether from birth or later on.
Yes, I think it's a really good question. And in my view, that is the counsel of perfection. I think that's right. But realistically, it's not possible at the moment. We just can't do it at the moment. We don't have the resources, we don't have the manpower, we don't have enough people with the right skills to interpret the genetics. We're very lucky where we are. We've got a group of people who know how to do all this. So I'm just a figurehead, really. I'm just, you know, I'm the person who's giving you this information, but there are lots of very clever people who are doing all the work.
Yeah.
There aren't enough of those people. But in a way, this is a bit like it was with scanning. When we first started scanning, so when I first started in epilepsy, that's what I was working on. And this is 30 years ago now. And then we could only do a scan for 2 people a week in London, in the whole of London. And you had to be really ill with your epilepsy to get a scan.
Mm-hmm.
Now, as you probably know, a scan is much more routine for many more people with epilepsy. And I hope the same thing will happen with the genetics. With enough support, then I think exactly what you say should happen. That becomes part of the standard.
But it might eventually sort of sell itself if you can treat, if you know from the beginning what treatment people need, you can give them the right treatment.
Absolutely right. Absolutely right.
Yeah. Okay, thank you.
Okay.
Thank you. Can we go back to Walter?
Yes, thank you. My question is in a similar line. What are the criterion for the selection of these drugs? Is it their internal structure or just the fact that it actually works by fitting in the gene?
So it's different for every individual case. There isn't a sort of template for it, if you like, because the different gene problems that cause epilepsy are all very different. And some of the genetic changes do what I've shown you today. They make proteins more or less active. Some of them cause proteins to fold in the wrong way and then gum up the works inside cells. And depending on what the problem is, you need a different drug to deal with it, a different type of drug to deal with it. So there isn't a single answer to your question. That's what we're finding as we understand more and more about more of the epilepsies. We see how complicated they all are. It's not one condition. It's a whole group of lots of rare conditions.
We have a question in the chat from Sharon, who asks, what exactly is gene therapy? Is it identifying genes only or treatment to rectify the missing gene or mutation? And can you see a time when genes could be replaced?
So again, a very good question. And again, there isn't a single answer, unfortunately, for most of these questions, because it depends on exactly what the problem is. So in some cases, you can, not in epilepsy yet, but in other conditions, you can replace the gene that's not working properly. You've probably seen some very high profile cases of degenerative eye diseases being treated by genetic therapy. In a way that's easier because you can get to the eye more easily than you can get to the brain. And the eye, the bit of the eye that you need to treat is quite small and very thin. In the brain, it's much more complicated. So we're not there yet, and it will depend on what the exact problem is. Sometimes it might be possible to replace a missing gene, if you like, or to put in another copy that does what it should rather than what it shouldn't. It will all depend on exactly what the problem is with the gene, if the epilepsy indeed is genetic for that person.
Thank you, Sanjay. And we have another question from Saima Aziz, who says, has there been any research done regarding epilepsy due to adverse reactions to vaccines in childhood?
Well, that's also a very good question, lots of very good questions. So the answer is yes. And in fact, that exact question was the reason why I'm doing genetics in epilepsy today. So when you're working as a doctor, it's often individual people who change your view about medicine and the problems that individuals have that make you think in a different way about things. And the very first person whom I wondered about a genetic cause and that set me off on this journey was somebody who was in their 50s and had bad epilepsy and walking and talking and learning difficulties. And he was losing the few abilities that he had. And this was all put down to the fact that he'd had a lifetime, 50 years of bad seizures and all its consequences with falls and head injuries. And his epilepsy was said to be due to a vaccination. He was said to have vaccine encephalopathy. And we had actually the very original letters from more than 50 years ago that said this young child who started having seizures after a vaccine, At exactly that time, some very nice science was done in a different part of the world that showed that many people who were thought to have a vaccination-related epilepsy actually had an underlying genetic epilepsy. And the vaccination and the inflammation and fever that caused, and many of you might have experienced that when you got vaccinated with for COVID. It often, you know, in flu vaccine, they often cause a little bit of a reaction. They make you feel a bit unwell, maybe give you a little bit of a fever. For people who've got that particular genetic condition, having that fever brings out their seizures. It doesn't cause it, it brings it out. And so that's a condition called Dravet syndrome, a really important condition to know about, because actually there are good treatments that you can use for people with Dravet syndrome. If you know what the cause is, then you can use those treatments. So the answer to your question is many cases of vaccine encephalopathy or vaccine-related epilepsy are actually due to an underlying genetic cause. Not all of them, but many of them.
And we have a question from Karen in the chat. Is there a waiting list for the genomics department?
So that's a very good question. As one of the questions asked earlier, can we do this for everybody? The answer right now is no. The resources just don't exist. And the way we do this is we see people who are referred to us, and we think about whether they might have a genetic cause. And if we think that might be the case, then sometimes we can get a genetic test done for that individual. But right now, we just can't do genetic testing for everybody. And that's one of our aims at the Society, to try and get that as routine practice for everybody. So the way it works right now is that you're referred, and then we have a good look at the history of that person's epilepsy. And if it seems relevant to do this and we can do it, then we will. But we can't do it for everybody at the moment.
Thank you. I think we've got time just for a couple more questions. I'm aware that time is running out. We have a question from Gillian Nicholas, who's asking, was the drug used in Charlie's case in use for any other illness, or was it by pure chance it was discovered to have the right impact on this protein?
Again, a good question. So it is used for another illness, and it was known to have an effect on proteins like this protein. And therefore, it was tried in the dish because a similar protein reacted in the right way to this drug. We thought, well, maybe this protein will as well. And we tried it and it worked. But I just want to emphasize that that drug is only for this very, very rare epilepsy. I don't want anyone to get the idea that this is a magic drug for epilepsy that will work for any epilepsy. It won't. And it would be, you wouldn't be able to get it for any epilepsy. It's a very specific drug for a very specific epilepsy. And I just used it as an example.
And we have a question from Claire Pelham.
Thank you, Sanjay. Really fascinating talk. I wanted to build on the point that Lucy made, really, which it is so wonderful to hear Charlie's story, but also rather poignant to hear about the gentleman that perhaps didn't have the opportunity to have the treatment that Charlie had at an earlier stage. And I know that 1 in 100 of us has epilepsy, but am I right in saying that potentially that means that there are hundreds or thousands of people, if you had the funding to reach them, who could benefit from genetic testing?
So thanks, Claire. Another very good question. I think the broad answer to that is yes, because I think, so what I've told you about is how the genetics can lead to the epilepsy itself. I haven't had time to tell you about how genetics can help with other aspects of looking after somebody's epilepsy, like telling you whether they've got a high risk of getting an adverse reaction, a rash to a drug, for example. So I think the answer is yes, I think genetics can help many people. That's not to say that everybody who's got epilepsy has got a genetic cause for their epilepsy. And again, I wouldn't want everyone to go away thinking everyone's got epilepsy, everyone who's got epilepsy has got a genetic cause of it. That's not the case. I was just talking about one aspect, obviously, of the science that we do. But overall, yes, it's a huge piece of information about each of us that we're kind of, not deliberately, but that we're not using, and we should be using.
Thank you. That gives us an extra incentive to shake that tin.
And I can see there's one more question from Alicia Iqbal in the chat about whether or not you take students for work experience at the research centre, Sanjay. That might be something that we can come back to, but I don't know whether you have a quick answer on that one.
Sort of. Yes, we do. There is a process, as you can imagine. So for everybody's safety, there is a process to go through, but yes is the answer in short.
Lovely, thank you. Well, I think we've just run over our time, so I think we're going to have to end it there. But can I thank Professor Sisodiya for giving up his time this evening to give us a really illuminating talk into genetics and health, and I think we've probably all learned a lot today. And thank you all very much for coming along this evening on when it's a nice evening when you could be sitting in the garden, and we really do appreciate your interest in the work that we do at the Epilepsy Society. And I hope those of you who are in Chalfont-St Peter will feel as proud as we do of the world leading research that we do.